It's my pleasure to serve as a modulator today. My name is Lingyun Chen. I'm a professor at the University of Alberta in Canada. My research focuses on plant protein structures underlying their functional properties. Thanks for joining us today for today's webinar to be presented by Dr. Yang Lan. Dr. Lan received his PhD degree in cereal and food science from North Dakota State University. Currently, he is a new postdoctoral research fellow at the same university. Dr. Lan has published 13 scientific papers in peer-reviewed journals and received 11 awards, including the IFT Graduate Scholarship and the AOCS Honored Student Award. That's a great achievement for young scientists. Dr. Lan's research focused on modification of parse proteins for improved functional properties through physical approaches, such as complex coacervation and solid dispersion technologies. It should be mentioned that Dr. Lan has become an AOCS member in Protein and Co-Products Division since 2018, and he has made many oral and poster presentations in AOCS annual meeting. Dr. Lan is also a leader in AOCS Student Common Interest Group. Now I will turn it to Dr. Lan for his presentation.
Thank you for introduction, Dr. Chen. Good morning, everyone. Thank you all very much for your interest to my study. My presentation topic is phase behavior of concentrated pea protein isolate pectin mixture and their application. A little bit introduction to myself. I received both bachelor's degree and master's degree of food science in China before 2016. Then I started my PhD training in NDSU cereal science. In past 4 years, I have published 13 peer-reviewed articles and gave 12 academic presentations at professional meetings such as IFT and AOCS annual meeting. I also received several graduate scholarship and honor awards from AOCS and IFT. Now let's move on to our presentation. Recently, yellow pea has earned considerable interest because of environmental sustainability, such as low water usage and low carbon dioxide footprint, allergy-free, and gluten-free that can be claimed in food labeling as an additional commercial value. The protein extracted from yellow pea seeds is cheaper than animal proteins and rich in niacin, which is an essential amino acid and very low in majority of cereal proteins. Therefore, a variety of pea protein products were commercially developed in recent years, such as pea protein isolate powder and pea protein milk beverage. In general, pea protein isolate, PPI, is extracted from yellow pea seeds using method of alkaline extraction followed by isoelectric point precipitation, AEIEP. Briefly, pea flour milled from pea seeds is dispersed in water at pH 8.5 and thereafter precipitating out protein at pH 4.5. The protein content of the PPI is commercially is commonly ranging from 78 to 86%. PPI primarily consists of 10 to 20% of water-soluble albumins and 70 to 80% salt-soluble globulins. Globulins can be further subdivided into legumin, vaseline, and convaseline. Moreover, Pea protein secondary structure consists of 75% of beta sheet and alpha helix. This characteristic composition and structure lead to limited pea protein functionality, such as low water solubility at neutral and acidic pH condition, which further limits pea protein commercialization, such as developing protein juice blend beverage. Polysaccharides are widely used in plant protein beverages for numerous reasons, such as improved protein functionality and food texture. In a solution containing protein and charged polysaccharides, biopolymers dominantly interact by electrostatic interactions via their charged functional groups. The interaction could be attractive or repulsive, mainly depending on factors such as biopolymer concentrations, charge density, or solution pH. Repulsive electrostatic interaction occur when both biopolymers in a solution carry charges with same sign, leading to phase behaviors of one-phase co-solubility at low biopolymer concentration or two-phase incompatibility at high biopolymer concentrations. In contrast, attractive electrostatic interaction take place where oppositely charged biopolymers mix in a solution. This interaction could be weak or strong, leading to the formation of soluble one-phase soluble complexes or two-phase complex coacervates. These two complexes are major interests of my study. Here are two applications examples of protein polysaccharide complexes. First, beta lactoglobulin pectin soluble complexes prepared at pH 5 can improve thermal stability of the protein by limiting the number of protein reactive sites. In another example, complex coacervates are used to microencapsulate bioactive compounds, linoleic acid, which could be easily oxidized. To begin with, core material, the linoleic, the oil is mixed with warm material, protein, and polysaccharide mixture to prepare oil in water emulsion at pH 7. Afterwards, the pH of the emulsion is reduced to certain pH to promote fabrication of complex coacervates. The liquid complex coacervates are then dried to produce microcapsules. As you can see, the powder particle is composed of core material of oil and warm material of complex coacervates. Finally, the oxidative stability of the microcapsules is evaluated. In this example, the oxidative stability of the oil is significantly improved. Traditionally, complex formation was performed at low biopolymer concentrations of below 0.3, and the turbidity measurement as a function of solution pH has long been applied to identify critical phase transition pHs for complex formation. Typically, this pHs, pH C by 1 and phi 2 were identified from turbidity curve during titration from neutral to acidic pH, corresponding to phase behaviors of co-solubility, soluble complexes, complex coacervates, and co-solubility again. Between pH phi 1 and phi 2, a maximum optical density was defined as pH OPT, which was believed to be the pH where maximum yield of coacervates were produced. The main drawback of this method is that for concentrated biopolymer solution, the initial turbidity is quite high, and turbidity increase abruptly. So critical phase transition pH as pH 5.1 and pH 5.2 cannot be identified. Also, it's hard to identify pH OPT due to the missing of a sharp peak like this. Pectin is an anionic polysaccharide and is mainly existed in plant cell wall. It has been worldwide employed in food industry as a stabilizer in acidic protein products. It is composed of galacturonic with galacturonic acid unit in the backbone. The carboxylic acid group in galacturonic acid residues can be methyl esterified to reduce charge density of pectin. Based on DE, pectin can be classified into high methoxyl pectin, the DE higher than 50, named HMP, with high overall charge density, or with low overall charge density, and LMP, the low methoxyl pectin, the DE is lower than 50, with high overall charge density. The sugar beet pectin, SBP, is a widely used HMP, since it has DE around 55%. Therefore, this study has three objectives. First, we studied how factors influence the critical protein polysaccharide complexes formation, PHS, at a concentrated PPI-pectin system by developing alternative methods to identify the PHS, including pH, biopolymer mixing ratio, concentrations, and charge density of the polysaccharides. Then we'll illustrate microstructure and physicochemical properties of PPI-pectin complexes, including soluble complexes and complex coacervates. Finally, we explored applications of PPI-pectin complexes in terms of improving protein functionality and microencapsulation of hempseed oil. First of all, we extracted PPI from yellow pea flour using AEIP method by dispersing flour at pH 8.5 and thereafter precipitating our protein at pH 4.5. Then we prepared PPI-pectin mixture by mixing PPI and pectin stock solutions, either the HMP, LMP, or SBP, at various weight ratio of PPI to pectin from 1:1 to 20:1 to study impacts of mixing ratio and charge density of polysaccharide. The concentration of PPI was fixed at 0.05 or 1 weight percentage to study impacts of biopolymer concentration. Subsequently, the pH value of the individual mixture was reduced from 8 to 2 by adding acid to study impacts of pH. Finally, the mixture was left to stand static for 24 hours to reach equilibrium. After 24 hours, according to different appearance of PPI-pectin mixtures, a state diagram was constructed as an alternative method to identify critical phase transition PHS. The phase separation was classified into 5 groups and labeled with these 5 symbols, namely the circle to represent turbid solution without precipitation, and the square to represent turbid solution without precipitation corresponding to the pH C, showing formation of soluble complexes due to weaker electrostatic interaction. The solid triangle, square, and circle representing precipitation with a cloudy or clear solution, representing formation of complex coacervates. Both zeta potential of biopolymer mixtures and viscoelasticity were determined and used as alternative methods to identify critical phase transition pHs, especially the pH-OPT, since the maximum yield of complex coacervates is typically achieved at 0 lead surface charge of the mixture, and there is maximum optical density, strongest attractive electrostatic interaction, and highest viscoelasticity at pH-OPT. Additionally, we elucidated thermodynamic properties in different phase behaviors using isothermal titration calorimetry, scanning electron microscopy, and further elucidated the long-covalent bonding of PPI and pectin using FTIR. In addition, for microencapsulation application of complex coacervates, we applied PPI-SBP complex coacervates to encapsulate hempseed oil, HSO. We had these 6 formulations. The core material is HSO, and the raw material is complex coacervate. There are many 3 steps to prepare microcapsules. First, we prepare HSO in water or emulsion by blending oil with PPI and SBP stock solution, followed by a homogenization. Then we reduce the emulsion pH to induce formation of complex coacervates. Finally, we converted liquid complex coacervates to microcapsule powder using a lab-scale spray dryer. Then we analyzed the physicochemical properties of microcapsules such as microencapsulation efficiency and microstructure. We also tested oxidative stability of microencapsulated oil. In this presentation, I will focus on oxidative stability of microcapsules, and you could ask me more information on other results later on if you are interested. Now let's look at our first results. How pH and mixing ratio influence phase behaviors of PPI and pectin mixture. The phase behavior of the mixture as a function of pH, mixing ratio, and pectin type were recorded and constructed as state diagram. For PPI HMP mixtures at mixing ratio 5:1 at neutral pH, the mixture presented translucent, indicating phase behavior of co-solubility. As the pH was further reduced to acidic conditions, mixtures presented turbid solution without precipitation, representing soluble complexes. The further decrease in pH triggered formation of complex coacervates with different phase behaviors. As I stated earlier, these markers correspond to these markers and here represent different phase transition pHs as I listed here also. Although phase behavior of precipitation and clear solution, the solid, square, in the state diagram was comparable to the pH OPT as I determined by a turbidimetric method. However, the real pH OPT means the pH for maximum electrostatic. Okay, sorry, what's trouble? Use. I think everything is good right now. Let me continue. So, as I mentioned, although the phase behavior of precipitation and clear solution, the solid square in state diagram was comparable to the pH OPT as determined by turbidimetric method. However, the real pH OPT means the pH for maximum electrostatic interaction, which cannot be determined by state diagram. When it comes to mixing ratio, the pHs shifted to higher pH values as the mixing ratio increased from 1:1 to 20:1. How about the influence of polysaccharide charge density? As you can see, the LMP used in this study has higher overall charge density than HMP due to higher percentage of unesterified carboxylic acid group in LMP. This difference led to earlier initial electrostatic interaction, namely the higher pH C and pH 5.1, and lower dissolution pH, the pH 5.2, between PPI and LMP than PPI HMP. When it comes to influence of biopolymer concentration, comparing with a low PPI concentration system, for example, 0.05 weight percentage of PPI, the pH ranges for soluble complex formation at concentrated protein condition. Here is the example of 1 weight percentage of PPI tended to be lighter, whereas the pH ranges for complex coacervate became broader. In addition, the critical phase transition pHs shifted towards higher pH as PPI concentration increased from 0.5 to 1 weight percentage. Here are zeta potential of PPI-pectin mixtures as functions of pH and mixing ratio. As the environmental pH decreased from neutral to acidic conditions, surface charge of the mixture gradually increased from negative to positive values. Since the pH OPT for maximum electrostatic interaction cannot be directly determined by state diagram developed in the study, we developed this zeta potential as an alternative method to identify the pH OPT, since it was reported that at the pH OPT, the zeta potential of biopolymer mixtures approach 0, namely the IEP of the mixture. Therefore, the IEP as a function of biopolymer mixing ratio and pectin type were represented here. We found that as mixing ratio increased, the IEP values shifted towards higher pH values, which is consistent with a state diagram result. Here is viscoelasticity, which is another alternative method to identify the pH OPT. since it was reported that the viscoelasticity G' and G" can indirectly represent strengths of electrostatic interaction. Higher G' and G" represent the stronger electrostatic interactions between biopolymers, and the higher coacervates yield. Therefore, we measure the viscoelasticity for all coacervates presented in previous state diagram. For PPI-SBP coacervates, prepare at mixing ratio of 5:1. The pH OPT was confirmed at pH 3.5 because both elasticity and viscosity were highest at this pH. Although you could find coacervates prepared at pH 2.5 have much lower viscoelasticity as compared to the value at pH 3.5, which means the coacervates prepared at pH 3.5 should have more condensed structure than coacervates prepared at pH 2.5. I will show you their microstructures in later slides. The ITC was applied to reveal the molecular recognition by titrating HMP or LMP with PPI at selected pH to represent the formation of soluble complexes and complex coacervates. As expected, as formation of PPI-pectin soluble complexes, reaction enthalpy, the delta H, was quite low, around 10, indicating a weak electrostatic interaction between biopolymers in soluble complexes. On the contrary, the formation of complex coacervates exhibited much higher reaction enthalpy, delta H, manifesting strong molecular interactions between PPI and pectin. Additionally, this table shows thermodynamic parameters of complex coacervates at pH 3.5, which is calculated from the binding isotherm. The relative values of T delta S and Gibbs free energy delta G indicated a spontaneous interaction nature between PPI and pectin. Both hydrogen bonding and electrostatic interactions were involved in interactions. Here are microstructures of complex coacervates at pH 3.5 and 2.5. We could see structures were critically dependent on formation pH, and their evolution could be described in two stages. To begin with, when at pH 3.5, the neutral aggregates of P protein and the hydrophobic interaction strengthen the complex coacervates between PPI and SBP. Thus, the network structures of coacervates become more condensed and much less organized. At a later stage of coacervation, when pH at 2.5, low neutral aggregates of P protein are involved. As such, the network structures are highly organized with relatively homogeneous large pore size distribution and smooth inner pore surfaces. This difference in microstructure tells us as for application of encapsulation, coacervates obtained at pH 3.5 and 2.5 may have different encapsulation results. FTIR analysis was applied to further examine non-covalent bonding between functional groups of PPI and pectin in soluble complexes and complex coacervates. I think from this figure, the spectrum of PPI exhibited absorption band of OH vibration, absorption of amide 1, 2, and 3. The spectra of PPI-pectin mixtures were overall dominated by the P protein due to the high PPI to pectin mixing ratio of 5:1. The significant change of the mixture spectra could be found in the carbonyl amide region. For instance, or mixtures exhibited the shift in peaks of amide 1, 2, and 3 towards higher wave numbers as compared to the PPI spectra. These changes indicated that both soluble complexes and complex coacervates were formed through the electrostatic interaction between the amine groups of P protein and carboxyl groups of pectin. At last, let's see two applications of PPI-pectin complexes. First, we evaluated the functional properties of PPI-pectin soluble complexes for improving PPI functionality. As seen from this picture, the PPI showed minimum solubility around pH 5. The formation of the soluble complexes, the ratio 1:1 to 20:1, represents the complexes. So the formation of soluble complexes shifted the minimum protein solubility to more acidic pH values. In addition, over pH 4 to 6, the formation of soluble complexes at a ratio 1:1 and 2:1. So the top two curves represent the formation of soluble complexes in these two ratios can enhance protein solubility to over 85%. Moreover, the denaturation temperature of PPI was slightly increased from 85 to 87 Celsius degree, indicating the thermal stability of P protein was enhanced. For second application, we used complex coacervates prepared at pH 2.5 and 3.5 as raw materials to microencapsulate hempseed oil, the HSO. The reason to select these two pH values is due to their differences in microstructure, as I showed you in previous slides. Here is oxidative stability of HSO in microcapsules. The oxidative stability of HSO in microcapsules was evaluated by measuring the secondary oxidation products hexanol and propanol as a function of storage time. Generally, the longer lag phase of the hexanol and the propanol formation is proportional to higher oxidative stability. As for impact of the pH, the microcapsules prepared from coacervates at pH 3.5. For example, here is F2. For pH 3.5 had a longer lag phase and therefore higher oxidative stability than at pH 2.5. So compare between F2 pH 3.5 to F5 pH 2.5. So F2 is longer than F5. Overall, the microcapsules prepared by complex coacervates at a wall-to-core ratio of 2:1 and pH 3.5 could be best candidate to prevent the encapsulated hempseed oil against lipid oxidation. So as for the ratio, so R1 means ratio 1:1, R2 means 2:1. So here we found the F2 has best oxidative stability. So that's why I conclude the pH 3.5 and ratio 2:1. To conclude, as for impact of factors on critical formation PHS, the pH shift towards higher pH values as the increase of biopolymer mixing ratio or concentration. High overall charge density contributes to higher pHc and pH 5.1 and lower pH 5.2 as compared to low charge density. Alternative method to identify the PHS for concentrated biopolymer solution. The PHS, PHC, pH 5.1 and 5.2 could be identified using state diagram, and the PHOPT could be recognized at the zero net charge of mixed biopolymer solutions or at the highest viscoelasticity of biopolymer mixtures. As for structure and properties of complexes, the smooth surface with homogeneous pore size could be formed at the late stage of coacervate formation when the pH is near the pH 5.2. The example in this study is pH 2.5. Both soluble complexes and coacervates are formed through hydrogen bonding and electrostatic interaction between amine groups of PPI and the carboxyl groups of pectin. As for application of the complexes, formation of soluble complexes could shift minimum protein solubility to more acidic pH values and slightly enhance P protein thermal stability. As for microencapsulation, coacervates formed at PHOPT, the pH 3.5, and wall-to-core ratio of 2:1 contributes to best oxidative stability of microcapsules. My work has been published as these 4 papers. If you are interested in my work, welcome to read them. That's all my presentation. Thanks for your listening. I'm ready. Sorry, I'm ready to take your questions.
Okay, thanks, Dr. Lan, for this nice presentation. Now it's the time for the audience to ask you questions. You can type your questions in the question box. Then I will let Dr. Lan know, and then he can orally answer your questions. While we are waiting for the question from the audience, maybe Dr. Lan, I will ask you the first question. Okay, so Dr. Lan, when you do the analysis of your coacervate rheological properties with rheometer, I see for your samples, for example, at the pH 2.5 and 3.5, you have 2 phases. I mean, one phase is just the liquid water. Another phase is the coacervate complex. So when you do that, did you analyze the whole mixture, or you just isolate the coacervate phase and to analyze their rheological properties? How did you do that experiment?
Yeah, thank you, Dr. May I ask a little bit about question? You mentioned I measure the rheology of the coacervates. So they are both complex coacervates. But you mentioned I measure the rheology for liquid water. I didn't understand what do you mean liquid water?
Okay, so if you go to your slide to show the rheological properties.
No problem. Okay, I think it's here. Yeah, this one, right?
And you also have, maybe before that, you also have a picture to show that, yes. If you look at the right side of your slide, you will see actually at the pH 3.5 and 2.5, you have two phases in the tube, right? So you have a phase that is the liquid, just the water, transparent water, and you have another phase, second phase, is the coacervate mixture, right?
Yes.
When you do the rheological measurement, did you measure the whole mixture, or you just get the coacervate part? You remove the water and just get a coacervate part, and you do the rheological analysis.
Yes.
How did you do that work?
Okay, thank you so much. I catch your idea. So just like this picture shows, so the left two test tubes here, pH 3.5 and 2.5. So they are the original the complex coacervates, and I gave them centrifugation for like 4,000 RPM for 10-15 minutes. So then you'll see this precipitates here. And after I test, before I test, I think these two pictures come from the end of the rheology test. So you'll see I only test the rheology for the coacervates. So I remove the water, the supernatant.
Okay. That's then. But while we're waiting, and for your other rheological measurement, if you go to the next slide, and you also measure the rheological properties at 4.5, I noticed that it's just one phase, right? For example, at pH 4.5, just the one phase.
4.5. Okay. Yeah. So for example, here you see this picture. These are these samples. So the picture I listed here, they are coacervates before I gave the centrifugation. So you see the upper phase is quite turbid, and of course there is some just compared to the pH 3.5. So pH 4.5 is a transition state. So there is coacervates and also the soluble complexes. So after give a centrifugation, definitely will so some soluble complexes can be transferred to coacervates. So of course I will also remove the soluble complexes, only test the rheology of the coacervates.
Okay, thank you for that explanation, because I see certainly your polymer concentration in these two scenarios, they are totally different, but you let me know you do the centrifuge for the samples. So I assume you have similar polymer concentration for rheological measurement. Thanks for that.
Yes, thank you so much.
Any question from the audience? I think you can, there's a question box in the toolbar, and you can enter your question here so we can have a communication with our presenter. Okay, so while we are waiting, I still have another question to Dr. Lan. In your slide, I think you mentioned the ITC result. Basically, that's technology to test the interactions between your pectin and your P protein, right? Could you go
Yeah.
to that slide? I think it's slide 22, likely.
Yes, I'm moving to there. I think it's this one. Yeah, it's this one, right?
Yeah, I think this is kind of quite interesting. If you look at the binding consistent in the table on the right bottom side, we notice that if it's high methyl pectin, we have stronger interactions with protein as reflected by higher binding consistent. Whereas if we use low methyl pectin, we have less interactions with the protein, right? And I think the first column N is the binding number. How would you explain that? Why with low methyl pectin, the interaction with the protein would be weaker compared to high methyl pectin?
Thank you for the question. So here, okay, I see there are two parameters. One is binding, the stoichiometry and binding constant. So here I'm thinking I think for H and to compare HMP and LMP, the interesting part is the binding constant. Not binding constant. It's binding. So here the 3.74 mean compared to like 1 means that the 3.4 units of protein to bind to 1 unit of LMP. So for HMP means 1 unit of protein to combine with 1 unit of HMP. So here, so for this one, it's quite reasonable because the LMP has has more unesterified carboxylic acid group. So that's why this value is higher than HMP. As for the binding constant, yes, I agree with you. You mentioned the possibility the LMP should be stronger than HMP. But here I have to see. So you'll see the significant difference. There is no significant difference between them. And as for the binding constant, I suppose I suppose the reasonable explanation to this is the binding and stoichiometry parameter. As long as this one is correct, I think this one it's fun to note significant difference between the two pectin. How do you think about this, Dr.?
I think numerically, from the binding consistent, it seems that interaction between the protein and high methyl pectin seems to be higher, although they are not significantly different. It might be because the binding number mainly determined by the charge on the polysaccharide chain, but the interactions, in addition to electrostatic attractive interactions, you may have other interactions at the same time. So for example, hydrogen bonding, other interactions. So possibly with high charged polysaccharide, you may be more difficult to develop other interactions. So I don't know the answer, but it seems to be interesting to me.
Yes. So let me take a look to the charge. So you mentioned the charge density. For example, I'm looking for the HMP and LMP at the pH 3.5. So HMP is the blue curve here, and this in here is I think here the blue curve is HMP, and here is LMP at pH 3.5 is -20. And for this one is around -30. But Also, yeah, so there is difference, and as you mentioned, LMP is has higher lead charge than compared to HMP. So you suppose the binding constant should be higher in LMP compared to HMP, right? But you also mentioned that,
But
yeah,
I don't know, but it's interesting to have further investigation. Here now, Dr. Land, I have some questions from the audience now. Okay, so the first question from Dr. Binchan Chen. Thanks for your great presentation. I have a quick question. How would the food industry adapt your findings into their applications?
Thank you. The question from Dr. Chen. So as for the application in food industry, as I presented here, we show the two applications. One is for one is to improve for soluble complexes to improve solubility and thermal stability of protein. So this is quite important and applicable in food industry. So part of appropriate amount of pectin added to pectin added to pea protein solution could improve first improve the protein solubility. That means higher concentration of protein in food products. Second, because of the higher protein solubility in a more acidic pH condition, so the pea protein can be applied to the food products with acidic pH, like the juice protein blend. So another application for microencapsulation is quite useful, because the bioactive compounds like the essential oil, not essential oil, the essential just like the the active compounds. So my study shows the good application, good microencapsulation and good oxidative stability of the microcapsules. So this could be applied to microencapsulate the product's active product, at least the essential fatty acids. So that's my answer to Dr. Chen. Thank you.
Okay, Dr. Lan, we have more questions coming. Then there's a question from Hong Sun Yang from National University of Singapore. Very nice talk. I have one question about PPI-pectin interaction. It seems that your PPI might contain some minerals. Whether did they affect the interaction between PPI and pectin molecules?
Thank you, Dr. Yang. That's a very good question. I never think about. I think about your question previous a little bit. It's about salt. So I think the mineral, especially the sodium and calcium. But I'd like to say, okay, I could tell you the minerals, the major component in the PPI could be the sodium, and the other components we measure is almost very limited amount. So the sodium typically it's almost the half percentage of the minerals, like around 2.5%. So the influence of the sodium, how to say, I mean, I mentioned the 2.5 means it consists of protein content. So for my application, my protein solution is 1 weight percentage. That means one weight percentage times 2.5 percentage of sodium salt. So I think this how to say, based on their study that who studied the influence of salt concentration to protein-pectin complex formation, the sodium concentration in my study could be ignored. That's my answer to Dr. Yang. Thank you.
Okay, and thanks, Dr. Lan. There's another question from Jennifer Kimmel. He asked the questions about if you don't use your pea protein isolated from your lab, if you use some commercial pea protein, which has much less solubility compared to those in the lab, how do your result will change? Because like in the industry processing, normally they have sterilization and spray drying processing to generate large scale pea protein, and normally they show less solubility. So if you use those protein for your result, for your experiment, how your result would be different?
Yeah, thank you. So for this question is very interesting. So of course, at the very beginning, I plan to study this project using the commercialized the PPI, and we found the picture as I attached the low solubility. So that's a really big trouble to this study. So you ask me what about my result using the commercialized PPI? I would like to say I cannot start my study because the concentration is very low. It's hard. Like, okay, the sample that I requested from the company is around how much 0.1 weight percentage solubility. So before I start my work, I will of course dissolve the commercialized protein and give a kind of no matter its centrifugation or the filtration to remove insoluble matters. Then I will start my study. So of course there are lots of work. They use the commercialized PPI with very low protein concentration, like 0.05 weight percentage. They can do that like that, but that does not make sense to for the industry, right? For application. So that's why I used the protein extract in our lab. And recently, of course, we found a method. We did find a method to produce PPI using spray drying method, and the protein can have good solubility. Of course, for my study, I didn't use the commercialized, but our other in our research group of Dr. Rao and Dr. Chen, somebody else, they are using the commercialized PPI and give the modification to them, and they also found good improvement in protein solubility. So that's another interesting study. Maybe in future somebody can introduce their work. Yeah, thank you.
Okay, thanks, Dr. Lan. Due to the time's sake, it's the time to wrap up the seminar. Thank you so much for Dr. Lan's nice presentation, and also thanks all the audience for your interest in this topic. Just let you know, the webinar, recorded webinar will be sent to all participants within 24 hours. So with that, wish you have a good day.
Thank you, Dr. Chen.
Okay. Thank you.